Let's get started so today is the second time of our cellular principles for biomedical engineering. Share the screen. So raise your hands if you can see the screen well enough. Yeah. Okay, great. So let's quickly go through some of this part, and then let's focus on your interesting questions for this question. So cellular principles in biomedical engineering. Today is the second part. So we will go over cell cycle, proliferation, cell differentiation, stem cells, and cell death and cell culture techniques. Okay. So I guess you've watched already things, and so I'll just go through with you. So in fact, there were questions about how the cell differentiation starts. And I think this is a good diagram that I think we already discussed it. So what are the decision-making factors for the cell to do something? For example, changing gene expression, or cells start to divide to proliferate. So when and why and they do that. So that's a kind of decision making can come from both inside and outside. So cellular proteins as biological clocks, special protein to promote inhibit cell cycle. This is a quite interesting things that I remember that some people to study how the cell kind of undergo a cycle and figuring out some molecules which seem to be also the cyclic pattern. So that interesting factor is found, and of course you have to name it. So how would you name it? Is the thing. So let's see. Youngmin, can you guess what this this factor, which is kind of relevant to biological clock? Can you guess?
Cyclin.
It's very close. Yes, cyclins. Okay. So for example, like there's a CDK cyclin dependent kinase. So like these are termed the cycle. So sometimes you will find the naming is not that difficult. If you know how the name was made out of that meaning, then of course the cells are not only like single entity. Like human, we are social animals. So we can't live alone in general. So we do have to interact with our families, our friends, and those affect actually our growth and eventually our destiny. That's why I guess you guys are on this school, you're admiring, and you're surrounded by, your colleagues and students and faculties, and that effect actually in fact nurtures you and growth for you so that you can develop yourself for a better future or maybe contributing to the world. So that kind of outside of influence is coming for this cell. There are like even growth factors such as epidermal growth factor or VEGF. Like those who are involved in interest in vessel, you should know about this called vascular endothelial growth factor, because this is something that causes a angiogenesis, especially for cancer development, or even your development, you have to grow your vessels. So VEGF is a very important growth factor. And that's outside that bind membrane receptors and that trigger to cascade, signaling cascade to express growth promoting gene. Okay, and the last one here is telomeres. So the name looks like telo is at the end. Okay, telomere is responsible for all overall lifespan and have role in cancer therapy. And telomere was discovered by, I think, John Sostek and Elizabeth Flatt. I forgot that actually the name. She is a UCSF, and they actually found this first time, and they realized that this is why the cell, if you grow normal cells, let's say primary cells from the body, they kind of divide. If you give growth factors, divide. At some point, they actually enter into senescence and they don't divide anymore, and they die. So you realize, oh, what causes this? And this is our humans like kind of thinking, if you can make the cells keep dividing until like without any limit, you can actually consider, hey, maybe I can reproduce my cells, my life, and I don't get age with that. So it could be very interesting. In that regard, our cells are programmed that we have something like this telomere, which is at the end of chromosome. Whenever you divide, the length of the chromosome, it's shorten and shorten and shorten. And in the end, when you divide, this exposes the end of the chromosome and the cell undergoes death or they can't divide anymore. So this is a remarkable discovery that actually gave a Nobel Prize in 2009. And what's more is interestingly, you guys know about cancer, the cells grow and divide, like keep dividing to make a mass, we call it tumor. And when the tumor gets bigger and that can be affecting our normal physiology and function. And also they actually kind of metastasize. Eventually that can cause like killing the patient. So in that regard, well, you can ask question, why the cancer cells can divide like without any limit? Oh, it turns out that 90% of cancer cells express enzymes. They actually add up more telomeres so that the telomere doesn't shrunken down to the end. So that can be an interesting target for cancer treatment. Because of this potential, they got Nobel Prize. So that, what are the name of enzyme? Let's see, Doreen? Can you guess?
Telomerase?
Exactly. So enzyme finishes as ACE, so telomerase. Okay, very good answer. And cell proliferation, and we've heard in previous high school, middle school class, we heard about G0, and which is called the gap. 0, so G stands for gap. What kind of gap between this is because it's mitosis versus synthesis phase. So S phase is called DNA synthesis. And this M phase is called mitosis. Mitosis is for somatic cell division versus there are different germ cell division. It's meiosis, you guys know. So there were questions about how do we know about like this phase. I think we'll discuss that afterwards. So the cell differentiation during embryonic development is very important in development and developmental biology. So I guess think you guys already have seen this. So let's see, Anho. So what are the diploid cell from two haploid gametes? So this is combining from sperm and egg. It forms a what?
Zygote.
Great, it's a zygote. And then when they grow, and mammalian blastula, it's like kind of this this shape with some differentiation is called
Blastocyte.
the blastocyst. So that cyst is kind of a cell, okay? And okay, here it is. So the question is, like we have developed, differentiate into various kinds of cells. Are all DNA in different cells in our body the same or not? Let's say, Yundami, can you guess? Ami? So all different kind of cells, do they have the same DNA or not?
Yes.
Oh, great. Yes, that's very important. We all have this, our cells have the same blueprint. And the question is, what makes all these cells different when they differentiate into different kinds of cells? So you can see ectoderm, germ cells, ectoderm, which is external, mesoderm, middle layer, endoderm, internal layer. So some terminology, I guess you guys already have seen. Let's see, Yundami again. So what are the outermost of cells of an embryo in early development? Tami?
Endodome.
Oh, actually this outer side, outside.
Exodome, I'm sorry.
You know, when I ask, usually I'm not asking too difficult questions though. Meso is what you say within is endoderm, right? And then the zygote, which already Anho is saying, diploid cell from fusion of two, which is a haploid gamete. So haploid means that DNA or chromosome is a half. Okay, so that they combine together to become a diploid, which is the next. Okay, so let's get into directly a stem cell, because all these are like from cell biology. I think you've seen it before, but stem cells are still compared to another part, it's relatively new, and it's in fact it's most exciting in terms of medical engineering point of view, right? So we have to define what are the stem cells. Stem cells are on, let's say, 정우주.
Specializing.
Exactly, yeah. They are not yet specialized, and precursor is kind of a pre-stage cell to become a fully functioning and differentiated cell. That can differentiate into diverse specialized cell type, or interesting stem cells can self-renew. That's why it's quite important. At the same time, you have to also be worried in some aspects, because if you make the stem cell a wrong way, they can become a tumor. In fact, the presence of tumor is a proof of, hey, this was actually stem cell, right? So why are stem cells interesting in BME? Their potential use in repopulation and tissue regeneration. I think it's so easy. Potential cell source for also cell replacement therapy. So when we from some disease or injury, if we lose a cell, we kind of have to replace cells. How can we do that? Well, stem cells might be a good for that, because it actually self-renew, and also seed cells for engineered tissue. So sometimes we engineer in tissue engineering. So let's say someone lost their ear, we need to reproduce or regenerate their ear. With a scaffold, you need a proper chondrocytes and skin cells or many other kinds. Then you have to engineer for that. So you do need a seed cell for that. So that's why stem cell actually, it's a very rare, the discovery of a ways of so-called iPS induced pluripotent stem cells. The paper in 2006 in Cell paper, I think that only in 8 years, the guy who got Nobel Prize just so picked. Why? Because that poses a great potential for the use of stem cell for human medical purpose. So we already know that the stem cells are in vivo fertilized egg. They can be anything in our body, totipotent. And then it becomes a blastocyst inside the inner cell mass of blastocyst that can become multiple different kinds of different tissues. So that's why we call it pluripotent. And once we differentiate, the only thing you call unipotent. So I guess that's something we already talked. And let's say this has to be go quick. Janice? Yeah, so stem cells have various different names. So like omnipotent, or it can become a complete viable organ, such as fertilized egg. A sperm and egg become a fertilized egg. That we call what? Janice, can you guess?
Totipotent.
Great, that's totipotent. And then, but then become different into nearly all cells, such as embryonic stem cells. How would you call that?
Pluripotent.
Yes.
Pluri.
Actually, when you have simple form and plural form, so it's coming from there. And what are the differences? Because it become, somehow it cannot make a placenta because it's coming from placenta. So that's why they're different. So I don't know how important that is, but that's kind of the differentiation. And it become a multipotent stem cell, such as hematopoietic stem cell. You will hear a lot. Hemato means blood. So, inside blood, various kind of different cells, such as RBC, red blood cell, white blood cell, has a lot of different types, and all these are coming from hematopoietic stem cell. And where does we made this? This is inside a bone, okay? So that's multipotent, and once it differentiates to make only one type of tissue, that cells that we call unipotent. So So embryonic stem cells are it's a very good source, important stem cell. But it's isolated from as they say, embryo, and so it's the most potent stem cell. However, we all know that from the religious point of view, there are like issues for like this embryo could be considered as a human or not. Then from religious point of view, this become a big ethical problem. So another approach is, hey, can we actually take out cells from adult? Okay, from the tissue on adult organism. Hey, that's no ethical issue. It may be painful, but it's no ethical issue. So that's such as bone marrow, peripheral blood, it would be great. However, like artificially derived from a non-pluripotent cells, typically adult, like body tissue, so that's what we call somatic cell. And this is a kind of revolution. And Shinya Yamanaka from Japan and more originally like John Golder from UK have paved the way of this induced pluripotent stem cell. So why do we call it induced? Because Shinya Yamanaka identified from like 126 or 8 factors, they actually do painstaking work to see which factors may actually re-trigger, reprogram the already fully differentiated cells into the original cell status. And so this is a Nobel Prize winning words for the discovery that mature cells, such as somatic cell, can be reprogrammed to become pluripotent. So this is a big feat. I think this is an amazing discovery, but like we have to see how much this actually can go. Okay. Then you can ask question, hey, by the way, how come we didn't know about stem cell before? Where does the stem cell exist? But from our experience, we know that when you have a cut, somehow a few days when you're young, it grows back. When you're old, it's a little less speed, but it actually refills. So you kind of consider, okay, there are some cells which remembers and just to recover. I believe this is just amazing, such as embryonic development. This is also very amazing. So we realize stem cells are very small quantities in kind of all adult organs, very small amount, but they are very potent to like replenish and recover a damaged or lost, or even a normal cell turnover stem cells would be important. So for that aspect, there are like a characteristic of stem cells. Let's say not. So what kind of unique properties do stem cells have? So these are some diagrams, and stem cells, once it divide, actually one become a original stem cell, so it become there, but the others become like differential. So that's kind of called what? Not, can you guess?
Asymmetrical.
Exactly. Okay, very good. And so for that committed cells, we call progenitor cells. Okay, so it's kind of a ancient cell which will become like differentiate. We call it a transit amplification. So because they have to keep like multiply and amplify, and so that it become like differentiated cell. So the issue is the cells keep like renewing itself. So that's kind of interesting aspect. So that's we call limitless self-renewal. So this is very important in some aspect. And also that is a lineage, a multi-lineage, not again?
Differentiation.
Great, yes, right. So these are differentiating steps, and that somehow is a multiple steps, a multi-lineage. And eventually what does the stem cells potent is because it can repopulate the tissues of the recipient. It can be self-owned or it can be maybe other. So in the end, in a nutshell, stem cells, self-renewal and potency to actually become some differentiated cells. All right. So this is just an example because your blood is always very interesting and very important. We call it hematopoietic. So stem cell become to like lymphoidal stem cell, myeloid. Lymphoid means lymph node. and that become a lymphocyte. These are so-called you call white blood cells, and so that's one part. The other is myeloid. Myeloid means like kind of bone marrow inside. Okay, so these become, so from the bone marrow, your RBCs are being born. Okay, I think that's actually this side, erythroblast. Okay, myeloid stem cell become another different kind of white blood cell, monocyte, which is a before macrophage, so cell-eating cells, and granulocyte. The name comes from the granular, inside the cell cytosol, they contain granules, like small little dots, okay? And some become RBCs. I know there's a very interesting questions about RBC, and also another interesting part is to protect our blood to be leaking. So that's coming from the platelet for blood clotting. Okay, so that's and hematopoietic stem cells are actually, you can see it can become different kind of differentiation. So it's a multipotent stem cell. All right, so adult stem cells location is in this diagram. I mean, this is very interesting. So you will have a question about where do we have stem cell. I'll give you 1 minute to watch this, okay? All right, so look at that. So you would ask questions about different kind of tissue type. Where are the stem cells located? It's very interesting. And one thing very interesting here is people develop a new idea of, hey, then if stem cells you discover, you find a stem cell, can it become any cells in our body just when you grow it on top of this Petri dish? That is actually an interesting question because it will become important if you want to develop, let's say, some kind of specific T cells or antibody producing B cells. Can you just grow them into Petri dish and they grow? This is a question we have to consider and ask. In fact, for example, hematopoietic stem cells, their stem cells actually located in bone marrow. What if these cells are not in bone marrow in some areas? Probably you cannot expect they will properly differentiate. What about a follicular epidermis or skin? So that skin's stem cells reside at basal layer of epidermis. So at the end, the bottom of your epidermis, there is a stem cell to reside. So another example, for example, T cell. T cell, we heard a lot about this from HIV virus are infecting T cells. For our immune system, T cells are very important. So how do the T cells develop? And the T stands for thymus. That thymus is in your in the chest, right? So they develop inside the thymus. And what are the shape of the thymus? That has like three-dimensional structure from the beginning to the end for development, or what we call maturation. You need a specific three-dimensional and biochemical cues within this environment. So for that, they grow, develop, mature, and become a functional T cell. And that specific environment, so people name it. Hey, that's like a very important place. So we call niche. Some people say niche, but that's something microenvironment with specific anatomical location where the stem cells are found and they interact with stem cell to regulate the cell's own fate. So this is a kind of a concept which I know someone who made this name who are having a liberal arts major. So that's very important. You imagine, think about it and name it properly. But these are such an important, when you are start to have the question, hey, I have this pluripotent stem cell. If I give a proper factor, do they actually develop to make a functional tissue or a functional organ? But the organs has specific structure. So I think it's a challenge. Think about the liver structure. Think about the complicated, let's say, brain structure. Will they form exactly the same? I think these are some of the practical questions as an engineering point of view you should ask. All right. So the last is a cell death. This is what I named. It's does cell commit suicide or homicide? So do they cell by themselves or kill by themselves, or they are like killing each other? Okay. So a good example in our development, I think this is fascinating for our digit formation because it starts from here. You see this red color start to die out so that it eventually make a digit. So that's very interesting. And this is we call programmed cell death. Okay, so that programmed cell death is we call a apoptosis. Okay, and the way that they die is very neat and programmed. So they make a blob and they don't like burst into their inside things outside. Why? In that case, that's called actually necrosis, such as when you hit by a bat or like then you have a blue thing or bruise, that's a kind of good sign of necrosis because when it bursts, the inside of the cell cytosol is actually causing inflammation. Okay, because plasma membrane rupture, inflammation, which is of course not good. And this, on the other hand, apoptosis, they don't cause inflammation, but instead they calmly do this, and then nearby macrophage or nearby cells engulf the debris of this. So phagocytosis by macrophages. So in the end, you consider like how come our body is sustaining our body mass or liver mass, and that's kind of tissue maintenance. Always the cells are keep, some part of it are keep dying. So you need also replenish. So you need to also proliferate. So sigma proliferation by cell death will make, if it's zero, I guess that's kind of homeostatic. So apoptosis, it's named as a programmed cell death or cell suicide. And there are a number of interesting features you already went through. On the other hand, necrosis kind of cell murder, a form of fatal cell injury, okay, such as premature death due to injury, like trauma, or disease such as infection, or ischemia, when there's a blood supply is blocked, the cells, oh my gosh, I have no supply of oxygen and nutrient, they undergo necrosis, okay? And the way they die is actually more devastating because cells swell and burst, releasing their content. And that's bad because it triggers inflammatory response. Okay, so in the past we saw this kind of a interesting diagram, but now in modern days, so actually my colleague, a professor who are in KAIST, who actually made a company to take this image, which is very interesting, based on just the light and without any reagents, use of light and the phase, so you can actually reconstruct three-dimensional cell shape. Then what are the contrast agent? Oh my gosh, contrast is just optical density or refractive index, because the nucleus and the cytosol has different optical refractive index. Even some of these cellular organelles have different optical refractive index, and that can be actually calculated, and even you can make a 3D, a real-time movie. So these are showing that a nice, interesting apoptosis. You see the cells blabbing and debris is coming out, and another cells come in and actually eat up all this. This you can actually take an image in real time. So I think that's great. And the last one is cell culture techniques, which so raise your hands who have done cell culture before. Raise your hands, or currently doing now. Uju, are you not doing cell culture? Okay, so okay, who have not done cell culture? I just want to know like how much you have done some. Okay, good. Oh, where's not here? Okay, hey, not, I can't see you anymore. All right. So cell culture is a so-called we call in vitro cell culture. I'm sure you've seen this already, so I don't have to go through. Just one thing I want to point out, there are cell lines. Those who are working, especially cancer biology, cancer cells are immortal, meaning that they keep growing. So you can actually grow them, and then you can split them, and then you can regrow. And how? Like almost forever you can do. That's why we call it has a almost infinite lineage that we call cell lines. Okay. Okay. So that cells somehow manipulated by human, and they proliferate indefinite. And so we can actually grow them for long periods of time. So it's great for us to do study with cell line. And a good thing is in over the world, we can actually share our cell line with each other. There are companies which sell cell line so that what your results can be reproduced by others. Oh, that's great. And what are the problems? Hey, these cell lines, a famous one is the HeLa cells, right? That has been like over like decades and decades from originally from a like cervical cancer from a black woman from the US. And it's great. However, these cells only represent one cancer patient. So that's the limitation. We have to keep watching and understand the limitation as well. Also, these cells are so much manipulated that the thing you found out from these studies may not be applicable to real a variety of different cancer cell patients. Okay, on the other hand, so you can actually choose that, hey, I'm going to just take out the cell. I want to study, let's say, a cornea. Then you took out the corneal cells from a living body, and that we call primary cell culture. And what are the problems? As we learned, telomerase. And so they may not live a long time, okay, directly from a living. What a good thing, because they are more realistic. However, outside of the body is still is limited because it's not in vivo condition, but then may be more relevant than immortalized cell line. And what are the difficulties? Because they don't last for, let's say, days, weeks, months. It's hard to keep them in that way, primary. And the other ways, because you took out the cell from the body, it's not pure cell, because our body, all the cells are mixed together, such as a stromal cells, immune cells, and blood vessel cells. All these are many times, all fibroblasts mixed together. So it's sometimes very hard to make a clean experiment, even though these cells may be more relevant than immortalized cell line. All right. So how do we do that? And this is a simple diagram for, let's say, for example, let's say I want to study metastasis, which I actually did myself, so I can tell you how we did. So you want to study breast cancer to brain metastasis. So you took out the breast cancer cells out, and then actually I did dissection of the tumor, okay, these tumor cells, and then I use enzyme to digest, which means we kind of separate the cells, and then we culture them. These are originally cancer cells, so they grow very well. And then I inject into the animal brain again, and they are growing over there so that I can study as a model for brain metastasis. So that can be possible. So, but the dissection and there are various ways. You can use either enzyme, or you can use a fine like knife to chopping, or further dissection when necessary. But in the end, what you can do is this single separated cell growth, or you just chop it. So the cell chunk actually contains kind of tissue morphology by itself. So you can use that as an experiment to study. Or even more, let's say I want to study the brain, especially, let's say, hippocampus, which is relevant to the memory and Alzheimer's maybe. So in that case, you kind of chop the brain as a slice, and you can actually keep that and study them by itself. We call brain slice, that we call organ culture. So even that's possible. Right, the cell culture initial, like single cell culture, when you do it, so they have to divide. So 1 become 2, 2 become 4. So you can see this is exponential. So when you grow the cell, so x-axis is passage, y-axis you can see as a log plot. That means it's exponential growth. And in the end, the cancer cells or cell lines become transformed. Do you remember when I say transform? transformation before, it's a different term. Like when the bacteria transform into producing some useful protein for us, that's also transformation. But now I'm calling this, the cell become weird, and because it become a cancerous cell, immortal, that we call cell line, that we call transformation. But many in the real primary cells actually cannot develop cannot multiply forever, so they undergo into senescence or they go die, apoptosis, right? So that's why it's hard to work with the primary cell. And in equipment, so these are kind of engineering. How can we grow cells outside our body? So we have developed, and over decades we have kind of discovered what kind of growth factor would be necessary, nutrient. So you need the nutrients such as amino acid and vitamins you add, and also the cells need glucose as an energy source. The problem is so you want to give out growth factors. Usually we use a serum such as a bovine serum albumin or BSA, and unfortunately these are coming from the animal like body fluid, like I guess blood plasma. You don't know, we don't know exactly what's out there, but we do know they contain growth factors. So we use this, give 5% or 10% to promote growth of the cells of our interest. However, we do not know exactly what factors are there. So that's why I call undefined mixtures of growth factors, hormones, and proteins. So you have to know the limitation of this aspect. All right, there are equipments, a number of laboratories in our department, including Professor Chang Myung's lab. Our lab has cell culture equipment, and I'm sure, and you're interested, you are welcome to come and visit and actually have a look how this cell culture is being held, a biosafety food and incubator to make sure the cells are at 37.5 degrees. Why? Majority of our enzymes are set as the optimal for body temperature. So that's why, and also at the same time, proper CO2 for proper pH condition. And for that, we have a number of tools called T-Flask, and this is well plate. Why do we need a well plate? Because whenever we do in vitro study, we want to always compare with the control versus your experimental number 1, 2 condition, and also a number of another control. So whenever you do experiment, the most important thing is the baseline or control experiment. Without control, you cannot change and use the different condition to compare. If you can compare, your data is shit, okay? So always at these kind of experimental design, the baseline is the most important. Okay, so that's what I can say. And there are like a refrigerator for keeping culture media, like the fridge, and water bath. So for keeping the media, when you pour into the cell, you want to have like warm one. But honestly, when I work with a cancer cell, you don't even need to warm it. They're so strong. But if you're working with a tiny set of like primary cells, very precious, only a few cells, you better to make this water bath to make the most calm, a nice condition for the cells. So in the microscope, when you grow cells, you can see every hours. You use this inverted scope to actually observe. So that's your role every day. You watch your cells. And when the cells grow too much, then it's a problem because many times the cells have a contact inhibition. What that means is when they grow and they touch each other, they communicate, hey, let's not grow anymore because we are full, okay? And that we call confluency. So you can see this one become higher and higher. And once the confluence is reaching 90, 80%, they start to even die. Why? Because they grow outside of their original plate and they start to apoptotic state. So you don't want that state, so that in general, like 80, 70% of confluency, you start to split them out, okay? So that's how you do cell culture. So, all right, so I think I'm going to stop it here. Gosh, I think I want to spend more time with your questions. So, All right, so let's see. All together, cell cycle and proliferation. Would you ask interesting question? How to distinguish cells in different phases? So we do know that the cells have different phases, right? How do we know? So I like this question because you don't just take, okay, cell has differentiation, but how? I think that's a very good question. So I just brought one here. So this is kind of, I put that one into Google. So this is just an example how to answer. For example, you see identifying cells under microscope. I think, you know, typically under microscope, you don't see this. I think they put some staining dye so that you can actually see the nucleus. But look at this. There are like cell nucleus, you can see there are these chromosomes at different, and this one is even dividing. Okay, and these are like all different kinds of. So this one actually shows the different stages of the cell. And this is something I can see over here, but when you put the cells under your microscope, you won't be able to see it because cells are usually transparent. Okay, you do need somehow contrast mechanism. All right, so Hamza Sharif asked about biochemists working on methylene blue dye. Okay, methylene blue is a like deep dark blue color, which is used for example, like dermatology when they, or surgeons when they have to take out the tissue, you need to orient them. So you can also use methylene blue. What happens when these dyes or medicine enter to normal healthy cells? Is external medicine produce toxic effect? Okay, I think this is a fair question and concern whether when we use methylene blue. So for example, you can even inject into the human body. Actually, I don't know about the use of methylene blue and actually for human patient. So I actually checked that out. So here, methylene blue is a dye given injection into a vein. And so it actually is being used for human treatment. And of course, with this, we have side effects such as headache, vomiting, confusion. Oh my God, all the serious side effects. But if you give this under proper dose, limited dose, you will be fine. Okay, so but then our question: what does this methylene do to our cells? Okay, so it's actually for staining dye usually. So determine cell mortality. So, for example, for a sample, so for those who have done cell culture, we actually want to know, let's say, the cell viability. So what we do is, I actually, it wasn't methylene blue, some kind of dye. So you have a cell suspension, you mix with the dye 1-to-1, and then you put it under the microscope. What happens is the dead cells actually uptake this dye and have a this pigmental color, such as like this. But normal healthy cells will not have that staining. Okay, so why? Because the dead cells uptake and they can't process, but normal cells has an enzyme to decompose. So that's something it's written over here. That's why healthy cell can, is a countless. So using this technique, you can actually see the quality of your cellular sample, right? So why does the methylene blue stain dead cells? Okay, here's a little more mechanism. The dye is oxidized as colorless compound by a reaction which is only happening in living cells. So dead cells cannot do it, so uptake, and then it shows that color. All right, but so as Hamza asked question, is that like bad for us? Okay, so there are some reports that carcinogenic. Some of the study are genotoxic in 3 assays, meaning that it may affect the cell's genome, and then that toxicity may cause cancer. So that's something it's good to know. And there are like it's even this how do you stain cells with methylene blue? You can watch this movies, which is nice. All right, so I mean there are many questions and answers, so you can actually add answers like here. Anho asked about neurons don't have mitosis. why do we have still brain cancer? I mean, this is a very valid question, and I think Uju answers very well for this. In fact, Uju answered for this. Yeah, neurons, they don't divide in general, so we don't have neural cancer in general. Our brain cancer, most of the time, it's non-neural cells we call glia. So that's why glial cells give out, they do proliferate. So that's why there are majority of brain cancers have the origin of glia. What kind of glial cells are in our body or in our brain or in our central nervous system? There's the astrocyte, microglia, or oligodendrocyte. Like these cells are non-neuronal cells, but astrocytes do proliferate. So there is, for example, a famous GBM, glioblastoma multiforme. So that G stands for glial. So brain cells, usually neuron cannot give rise to cancer. I hope that makes good sense for you. Not ask what triggers cell differentiation. Where's not? Okay, there. So this is kind of an open question, and I like it. So what triggers our cell differentiation? So we can actually go type it into Google. God of Google can tell us anything. So I guess this is worth to look at factors. So some factors will trigger cell differentiation. For example, cell signaling, including, and that's inside. So the normal cells will trigger themselves. Hey, it's time to divide and make more of us, multiplication. Or sometimes environmental influence. Okay, and of course, in developmental stage, it is a crazy developmental differentiation stage, and of course, we are programmed at the level of development from the embryo to become a baby or organism, and even at the adolescent growth. Okay, so that makes sense. And the other is the cell signaling, I think is there. So what factors regulate our cell differentiation? And now we can actually tell more. Process of cell differentiation is regulated by transcription factors and growth factors. All right, so what others you can kind of consider as a cues outside of cell cellular cues, such as growth factor, and you remember cytokines. So cytokines may influence a immune cell differentiation. And of course, the cell-to-cell contact each other. Hey, some cells are missing. Hey, I lost my contact, so thus I have to differentiate to make another cell from the stem cell point. So that makes sense. Also, ECM or extracellular. So you guys all remember that when you're doing like sports or guitar, you have to make more of a thick callus in your skin. So why? To protect yourself, right? So then this mechanical force gives rise to signaling that, hey, I do need more thick skin here, okay? So those also mechanical influence also triggers the cell properly differentiate, and that's a physical force as well. So I think that's a very interesting open question. And Uju asked even more interesting, like which point, good moment to call it alive? Guess true love. So Uju's question, I guess, is about at what point can we differentiate a living? Probably we call the cells as a living body. So I guess it's, let me see. Uju, can you explain your question again? And what aspect did you ask this question?
Actually, I just wonder, in embryonic state, embryos are progress diploid, haploid like that, and when we can call it's the live state, I mean, we call baby.
Okay,
which point is?
Right. I mean, this is a very, very important question. It actually affects all our human society, in fact. Why? You can kill an embryo, you can kill cells. does people call you, hey, non-ethical or not? So because of this, actually, stem cell research has been controversial. And that's why also induced pluripotent stem cell got Nobel Prize because it resolved it. So the question for us is, from when we can tell a living organism, so then you have to actually define what do we call as an organism, as an alive. So there are a number of like characteristics you can find, characteristics of living and non-living things, such as breathe, eat, grow, reproduce, and have senses. On the other hand, non-living, do not eat, and do not reproduce, do not breathe. So does the embryo breathe or not? Probably not. And these are very much important to, it's on a debate that especially from the religious point of view, okay? Because the defining features of life as a internal growth, locomotion, do they move or do they reproduce? Do they respirate? Or do they respond to external stimuli? I guess that's very important. And so that's what we call as a, let's say, can we say a human being from this embryonic development? And there's some of the information here that about 9 weeks of gestation, can you say a human person after fertilization? And so the personhood, when the physical appearance of fetus remembers a mature human at about 9 weeks, does that mean that we can allow to kill 8 weeks of age? Can we allow to killing of it? So Youngmin, can you have your own consideration? Can we kill a human at 8 weeks of gestation because they don't form a mature human shape? What's your opinion about that? Oh, we can't hear you very well.
I don't know about this.
Yeah, then can you guess what actually would be a criterion that you can actually kill an embryo, whether because it's not a human being? Is it going to be the blastocyst or stem cell stage, or 1 week old, 8 weeks old, 9 weeks old? How would you give rise to your own criteria to justify even abortion? I mean, you should think of it your own ways too. Youngmin, no opinion?
Yes, I don't know.
Yeah, okay, it's fine, but I want you to think. I would say my own opinion. It's not the answer though, but I do have my own opinion. So I would say pain is the determining factor whether we can call it an organism, a human being or not. So if there's no nervous system, because when you do abortion, you have to chop the embryo and kill it anyway. When the embryo feel pain, I guess that's kind of non-ethical. So when the CNS, central nervous system, develops and they don't respond to external stimuli, if they don't feel pain, I guess that's probably the time when we call it ethical or non-ethical. My own opinion, I guess this is more like subjective, and we can make an agreement or non-agreement. But I do ask you think of this interesting question. Which point is good moment is alive? If it's not live, then we can actually kill it and we can use it for our experiment. Hopefully that gives you something to think of. Janice has a question. How can you bring stem cell for cell replacement therapy? Where can you bring the stem cell to be used in treatment? Very, very interesting question. And I think this is a practical question that, hey, there's a stem cell. So how are you going to use it? So I have a heart attack. So 1/3 of my heart cells are dead. How can we use the stem cell? And in fact, this is an active area of research because we realize you give a stem cells into the IV injection, we realize only less than 1% of that cells actually go to the heart and start to proliferating there. So the efficiency is always a problem. So the delivery is an issue. Local microenvironment is a big issue, even after the discovery of stem cells. So let's see, like stem cells. So for answering that question, stem cells which used in cell replacement therapy. So that's a question. We have, let's say, the potential source of regenerative medicine is human pluripotent stem cell, including iPS cells. So this iPS cells or embryonic stem cells, or even human neural and mesenchymal cells after transplantation into humans. For example, like neuronal precursors will contribute to behavioral recovery, especially with the PD or Parkinson's disease. So, you know, you always have to, when we do this kind of treatment, you have to think how far are we going? What are the ultimate goal? Is in fact here behavioral recovery or functional recovery. So you already shoot for or will be the most important determining factor or assessment in the end is going to be functional recovery, behavioral recovery. Okay, so that's something I want to make sure. But what are the current status of stem cell therapy? So for example, spinal cord injury, heart failure, retinal macular degeneration, our tendon ruptures, and diabetes type 1. Diabetes type 1 is there's no cells to produce insulin. Okay, so that's why you actually give a stem cell to produce insulin. So that can help, but honestly, I haven't heard so many. We do know a lot of development in this area, even many fake medicines, because there are a lot of restrictions as well. But one thing, famous one you can actually see of a stem cell treatment is the actual blood-borne hematopoietic stem cell transplantation, especially also I think a Nobel Prize winning discovery of bone marrow transplantation, especially for white blood cancer patient. Because of blood cells become cancer, you kill out all the bone marrow cells and replace it with a non-cancerous to treat certain blood and immune system disorder and rebuild blood system. And I think this is just a wonderful, and that's why bone marrow transplantation, a Nobel Prize out of this. All right, so hopefully I think that's something very relevant question. And Youngmin, okay, ask a question. When induced iPS cells is derived, I just love this question, okay, because we know that there are not only conventional DNA to RNA to protein during the process, so-called epigenetics is happening for the final functioning. And that's a little different from what we call stem cell. We didn't talk about it, such as methylation. And when induced is derived, does all the modification in DNA is happening. So Youngmin, that's your question, and I think you are the right person to explain your thought on it.
When industrially the stem cell is derived, DNA histone modification or DNA acceleration can change like prototype stem cell.
Youngmin, I just love this question, okay? Why? Because I haven't thought about this before, and I think I encourage you to answer your own question in the YouTube, or also welcome others as well. Why this question is very valuable? because we know that like from DNA to RNA and protein expression to make the final cells differentiated functional cells. But also we learned that there are not just a genetic aspect, but epigenetic aspect, which may modify the cellular differentiation, the final state, such as DNA methylation. So when we work with stem cell, do they actually also do this exactly the same way we do? This is a very important question. So I just encourage, I like this. So honestly, I don't have the answer because I'm not actually working on it directly, but this is fascinating question. I do encourage you to try to find out a simpler answer for that. Yeah. And hematopoietic stem cell. So here, Youngmin asked also a very interesting question. RBCs make a protein without the nucleus. So there are Janice and Uju put the answer. So I encourage you guys to read these answers, which is, I think, just fascinating. A good question, because this is very curious. You know, RBCs are a final product of this hematopoietic stem cells. And then they differentiate. Let's say, I think I have, let's see, let me go back to our, oh yeah. So here, this. So erythroblast. Erythro is just a professional name of RBC. So we call RBC on the other erythrocyte. Okay. So stem cell, hematopoietic stem cell into erythroblast, and they become RBC. Interesting, RBCs do not have any nucleus. So that means there's no DNA, no RNA, no proteins. But it does have its protein already inside. So I think RBCs kind of do not proliferate itself. So that's interesting. This is my opinion, and I think the others are put there, but I don't know whether it's really true or not. But in general, without DNA, and they lack nucleus, so they cannot express genes and a new synthesite. There's a lot of protein, special protein inside RBC, including hemoglobin, but I guess there's no more of that. That's my guess, but if you guys find out it's not true, that's wonderful, okay? And another question, Uju, do they reverse differentiation for RBCs can redifferentiate? So this is very good question. Can RBC be redifferentiated to erythroblast? Uju, can you answer your question? I guess you have some idea whether.
Actually, I have no idea, but I guess just maybe it can't.
So if RBC can be redifferentiated into their original erythroblast, how would that be possible? Oh, actually, this is a good question.
Oh, could you say that again? What was your question?
Yeah, so Uju, I think I like this question because just a simple thinking, hey, it's differentiated. RBC has no nucleus. Erythroblasts, but have a nucleus. So from nucleus to no nucleus, okay, that's what we see. Without nucleus, can it go back to the with nucleus? So a simple thought for me is probably impossible because the induced pluripotent stem cell, how do they make it? We have somatic cells. We give out proper transcription factors. They affect the gene expression of these somatic cells to reprogram their another set of original gene expression to go back to the stem cell. But RBCs lack DNA, so they cannot change their gene expression. So that's my simple thought. You can correct me on the YouTube if I'm wrong. But that's a very curious and interesting question. I think this is a very nice question. And Jenny Fuang asked the question, which part of adult body could we take stem cells safely, unharmfully for medical purpose? Oh my gosh, okay, that's a very, very good question. I encourage you guys to put down your answers. But my simple thought, we do know that like from bone marrow transplantation, we actually have to take out bone marrow stem cells by inserting a big needle into our pelvis. You know, our pelvic bone is a big source. So that's how we actually take out bone marrow cells, which contains a lot of stem cells, right? But that's harmful. Actually, it's very painful. That's what I heard. But for stem cell therapy, in principle, you can take out your skin cells, which is probably not that harmful. But I do not know from where in our body we actually take stem cell. But just one thing for genesis, for delivery, the baby has this cord, you know, cord, right? Your cord, that's actually discarded. So I do know that cord is being used for cord-based stem cell source. But I'm not sure whether we actually took out our actual tissue. So you are welcome to actually figure it out and put the answer. Can you use stem cell for recovering dead cells? Oh my gosh. Okay, so I don't know, like definition of dead cells, apoptotic or necrotic. Probably, I'm not sure we are reviving or we are resurrecting dead cells, because from our science point of view, a real definition of that is probably you cannot recover. It's my own view, but you guys can think of different. Anho asked a very interesting question, stem cells, because neurological disease is most devastating in this aging society. So it's a natural to ask this, can you use stem cell to treat neurological disease? Oh my gosh, okay, this is a wonderful question. And I think I did some search for that. So I'll share, I mean, this is just an example, but you guys are encouraged to do this. Okay, transplanted stem cells can migrate into nervous system and integrate into neural circuits to enhance, to improve synaptic transmission. Oh, so there are some studies for that. How stem cells used to treat neurological disease? So for example, another devastating one is spinal cord injury, right? You know, this is something people really, you know, become debilitated, cannot move their part of or the whole of behind, below the neck or below some part, which is devastating. So neural stem cells offer potential to replace lost tissue after nervous system injury. It said potential. That means we do not yet have very clear. Thus, stem cell can promote post-neural repair in part by how secreting growth factors, their regeneration promoting activities, which can be modified by gene delivery. So it's actually suggesting that proper genetic modification stem cells may help repair. So another stem cell therapy probably the only potential modality offered to cure for neurodegeneration. I mean, neurodegeneration is degeneration, so that means it's very hard to recover, right? So this is an opinion that, hey, stem cell is the only way for treating to cure. Other methods may not do the job. So this is quite strong statement, but honestly, I haven't seen much evidence for that. But there are, you can hear from Switzerland and kind of helping rehabilitating spinal cord injured mouse or rats, and then they show some. That gives a hope, but it's not yet in routine clinical practice. That needs a lot more research and even probably biomedical engineering. So recent studies have found exogenous stem cells can migrate to damaged brain. So the first thing, somehow stem cell has to be there. Remember stem cell niche. The cells need proper microenvironment to do their proper differentiation and their job, okay? Then participate in repair of damaged brain tissue by further differentiation to replace the damaged cell. and while releasing anti-inflammatory factors and growth factor, thereby significantly improving neurological function. So in the end, how can you judge that? Do they actually improve the function? Do we get functional recovery? That means you actually are really having impact for this medical treatment. So, okay, and Uju asked a question, apoptosis necrosis. Do dead cell or residues also removed by phagocytosis or other? I believe these are true, although I haven't seen with my own eyes yet, but these are true. But if there are anyone who have a new or different opinion, I would welcome to add to discuss this. Jong-Yun, if cancer cell has an injury, does the necrosis of cancer cell happen like a normal cell? Very interesting question. Jong-Yun, but can you explain what do you mean by cancer cell has a fatal injury? Can you bring an example or why you ask this question?
Recently, I saw in my cancer, I influenced some cell on, and there are they contaminated by microbiota. So so many of the cells died by the microbiome. So because in this process, the microbiome have did the damage to the cell, so it so this make this question.
Oh, okay, so you talk about this pipetting.
Oh no, no pipetting. So there are some the mycoplasma is some
Infection.
Like kind of a contamination.
Mhm.
Right. It can some damages the my experimental results.
Okay.
So
Okay, that's kind of relevant to the actual experiment. So honestly, when I cannot know the answer directly, it's a good question. The cancer cells, for example, when it has an infection, so you are talking maybe some mycoplasma infection, something, then they may undergo death, necrosis. Do they happen like normal cell? Good question, and I do not know the answer. So, so the cancer cells are also part of our cell. Okay, so that's part of our cell, but they have a wrong, for example, proliferating circuit that they keep proliferating, or they do have a lot of growth factor genes overexpressing that they keep having growth signaling. But in terms of apoptosis and okay, now I remember. It's a very good point. So some cancer cells do resist apoptosis. Do you remember the cell circuit? There's a fast ligand coming in because these bad cells are supposed to get the signal from outside. For example, some other cell, immune cells activate fast ligand. Hey, you guys, you should die your cell. The cancer cell develop to resist the cell apoptotic pathway. Very interesting question. If you are interested in kind of cell cycle, cell death, especially in cancer point of view, I think this is a great question. And honestly, I don't do research in deeper insight, so I can't answer. But it's a fair question, a very important question. So let's see, move on to last question by Uju. Is it possible we keep the stem cell phase without proliferation during keeping cell culture? Okay. So 우주, where's 우주? Okay, there. Keep stem cell phase without proliferation. Okay, so this one, another good question, which honestly, I haven't done stem cell research myself, so I cannot really answer for this directly. So I encourage you guys, Google search and figure out. One thing I can comment on this is, in fact, stem cell is very much like common with cancer cells. Why? Stem cell has limitless self-renewal. So the stem cells like growth, you can grow stem cell like a mass. So forming like a tumor like mass is a proof of whether it's a stem cell or not, which means stem cells are very much strong. They don't die very easily because you don't want to lose your stem cell. So that's something I guess we can keep the stem cell phase for a long time without proliferation. I feel this is how real stem cell in our body actually do. Because even you are very old, you still have to have stem cell to replace and repair your injuries. So I guess that's my guess, but I do not know because I don't do stem cell culture myself. So